Good afternoon and aloha. Welcome to our April Speaking of Health lecture series. My name is Eric Barsatan and I'm the manager for physician relations and medical staff services here at the Queens Medical Center, West Oahu. Today's topic is COVID-19 vaccination, process, prevention, and protection. Again, in the past, we held the Speaking of Health lecture series in person here at our Queens West campus. But for now, for everyone's safety, virtual is a new format and we'll continue to go with this format until further notice. Right now and throughout the presentation, if we could ask you, all our viewers, to be on mute. And if you have any questions, please type your questions in the chat box and Dr. Ronald Kuroda will answer them at the end of his presentation. At this time, I am honored and privileged to introduce our speaker. Dr. Ronald Kuroda is the Chief Medical Officer here at the Queens Medical Center West Oahu and the Medical Director at the Queens Transfer Center. Dr. Kuroda attended medical school at the University of Hawaii John A. Burns School of Medicine and completed his residency in emergency medicine at Orlando Health in Florida. He has been practicing emergency medicine physician at the Queens Medical Center since 2009. So Dr. Kuroda is on your screen, and all of you should also be able to see him in his PowerPoint presentation. Welcome, Dr. Kuroda. You may now proceed with your presentation.
Thank you very much, Eric, and good afternoon, everyone. Thank you for taking the time off on most of your lunch breaks to listen to the Speaking of Health that we're putting on here from Queens West Oahu. The whole idea behind these is to give the community and the people who work within our walls the opportunity to hear a little bit about some of the topics that we have that are timely. And of course, with everything going on right now, we are in the middle of our vaccine drive, not only here at West Oahu, but actually across the entire world. So the information that I'm hoping to give to you hopefully will allow you to get a little better piece of comfort about what actually has developed with vaccines over this last year where we've been dealing with COVID, and for you to be able to ask some questions and also communicate to your family members and your friends who might have some questions or might have some hesitancy. We do want to provide everyone with education and information. Of course, vaccination, your medical care is always your personal decision, and we do respect that. And we also appreciate the opportunity to talk with you and share some of the information about the vaccines, the technology, the outcomes, and what we're really trying to do. So what I will do now is I will start my presentation and share my screen. Okay. So the topic again is the COVID-19 vaccination, and I wanted to split it up into a couple different bite-sized portions for us all. I don't want to get too deep into science, and I'm a kind of straight shooting kind of guy, so I just like to bucket things. What I'm going to talk about is an overview of some of the vaccine development, because that was a very big concern for people. We heard a lot of concerns about the speed, the safety, were they skipping steps or cutting corners. The prevention, what we do in Hawaii, how are we using it to prevent disease from spreading? And Hawaii has been an excellent state from the United States as far as for keeping the number of COVID cases down. We also want to talk about the West Oahu experience. Since we do have a large population of people out here, this is where people live. And since a lot of us have been home because of the economy, we wanted to make sure that West Oahu was represented and that we delivered this kind of care and prevention out to our community. And then also, I'll end up with the science, some of the basic science about how the Pfizer vaccine was developed and how they tested it to make sure that it was having an effect as well as the safety in it. And we are pretty early in this COVID vaccination time, so there is not complete information yet, but I will provide as much up-to-date information as I can about the long-term reliability of antibody production. So first off, I just want to disclose to everyone that I do not have any financial relationship or sponsorship with any of the vaccine manufacturers. The opinions that I have and what I'm going to be communicating to you is information that is readily available on our public information websites, on the journals that are published from reputable medical journals and manufacturers. And our on-the-ground West Oahu experience. So we'll start off with vaccine development as an overall thing. This graph here shows that in the normal vaccine production and research that we're used to, it does take about 10 years to develop a vaccine that's workable, including the research behind it, the clinical trials, and then getting the authorization. If somebody does have a desire to develop a vaccine to address an infection, it usually does take a year and a half of what we call bench research, or the test tubes, to come up with a formula or a way to approach the vaccination production. Then when you get through those different stages, you come to this phase here where you'll actually start to test the vaccine or your product on animal studies. Then you'll come up with some data, and you'll then go to what's called phase 2 and phase 3 clinical trials, which is to do with testing in actual people. You want to make sure that the benefit and the risks are evaluated, and that the benefit is higher than any risks that were identified in the earlier stages. When they talk about factories and manufacturing, usually the phase 1 through 3 trials, you will not produce vaccines in a large scale. There is a cost, and if it's not a valuable or an effective vaccine, the previous way is that the manufacturers wouldn't front that kind of capital or that kind of risk for something that might not be able to be effective or come to market. Once that happens, then they would go for approval from the governing bodies and then to distribute them. When we're talking about flu vaccines, those previously had been distributed out to the clinical pharmacies, to the doctor's offices for widespread distribution. So this is the pathway that we've been used to. This box here on the left-hand side is actually a description of what happened with this COVID vaccine. It wasn't that steps were skipped, but what actually happened was that the steps were run at the same time. The research actually was pre-existing. It's not new approach to developing a vaccine. It's just that it really wasn't widely used for vaccine production. The preclinical stages and the phase 1, 2, and 3 trials were also done in a very quick fashion. What usually would happen would be at each stage, the manufacturer or the researchers would have to evaluate to see if the cost for producing the next stage is justified. But there were some worldwide initiatives that really helped cut those down. Another risk that the manufacturers did was they took this leap here, and they started building their manufacturing factories and capability at the same time as they were doing their phase 3 studies. As you can see from the larger graphic, usually that's done in a stepwise fashion, and people aren't going to be taking that risk prior to approval. This approval, when I get to the stage about talking about the FDA and the CDC, happened in the middle of the manufacturing process. So again, they were taking the risks that they would have a product that was going to be effective. And we're now in this distribution phase where it is going across the nation and the world. So to answer the question for folks, if they were worried about how quick it was developed and the question about where all the steps followed, this graphic kind of outlines that all the appropriate steps were followed. It's just that the timeline was condensed. So let's talk about the worldwide timeline. The first reported case of SARS-CoV-2, which is the technical term for the COVID-19 causing virus, was reported in a patient who developed symptoms in December of 2019, hence the last two numbers 19. Science was able to gene sequence the virus of the SARS-CoV-2 in January of 2020. That was a critical part for determining what the roadmap would be, what kind of targets we would look for. This little picture here is what we all recognize when we watch the news, Hawaii News Now or KITV, and they have this cartoon, a graphic representation of what the virus looks like. What the gene sequencing was able to do was figure out what the gene sequence or the signal was to produce these spike proteins that are characteristic of the of a coronavirus, and specifically what made the SARS-CoV-2 COVID-19 coronavirus so contagious, and what we attribute some of the severity of the infection to. So while this was all going on, a lot of the reports coming from around the world were coming in. This is an old graphic from Italy, and this represents March of 2020. It does show that even if we're starting in January, by the time that people were looking at it, they could see that this huge upward rise in Italy was very concerning, that this was a very contagious type of infection, and resulting in, even at that early stage, over 1,600 deaths. And it also impacted greatly the population that were over 65. And with that kind of information that was coming out and looking at this pattern, it did trigger the concern that a worldwide event, a worldwide infectious condition was coming. Because of that, it did spur a global effort. Other countries, pharmaceutical manufacturers, and it was motivated by that need in that last graphic, because we could see that if it was happening in Italy, and as history showed, New York City in the United States had a huge hit as well, the need to find something that could prevent this was necessary. We would not be able to treat our way out of it once people got the infection. There were other things, other measures that were going into place, masking, physical distancing, and a lot of that information was based off of our previous experiences with respiratory viruses. Prevention was the key, but because we would need to maintain our function in the world, we needed to find some other method, and vaccines were the natural source. Vaccines and their theories have been around since cowpox was used as an inoculant to try to prevent smallpox. And we were successful in the world of eradicating smallpox. And that is an appropriate use of medical knowledge to promote health and prevent serious illness in the human population. Other things, like I spoke about, was private and government funding. The United States was huge in talking to the manufacturers. President Trump, when he declared a national state of emergency for this, opened up a lot of resources of the United States government to fund research and development, and it allowed the manufacturers not to have to worry about having a failure and going bankrupt. They could take those risks and develop things that would benefit the overall world population. So as early as August of 2020, the pharmaceutical companies were coming up with the approaches that they were identifying as effective. The BNT162b2 vaccine was developed. They actually had two different varieties, the B1 and the B2 variety, and the B2 was most effective in their phase 1 studies. They continued to take that and then do the other stages in that graphic that I showed earlier of the phase 1, 2, and 3 studies. And then in December of 2020, this clipping from the New England Journal of Medicine published their findings. And this made news all over the world because it was the first publication of information showing the phase 3 studies. It allowed, because it was published, it allowed people in the medical community, and actually anybody who had access to the internet. The New England Journal actually published that study free of charge for anybody to take a look at. And in the scientific approach, we want to take a look at the information, see if it applies to our patients, our population, see if we can use it, and if the data and the conclusions make sense. What happened when that was released was the United States FDA took a look at the information, used the same process that they did, and critically analyzed the data that was published. And they did what's called an emergency use authorization on December 11th. An emergency use authorization is a little different than when we say something is FDA approved. However, the level of scrutiny and the information that they require to even get to that point is very valid. In the United States, we have different levels and different thresholds. The second body that had to be approving or authorizing the use of this was the U.S. Centers for Disease Control, the U.S. CDC, and they have a branch called the Prevention Advisory Committee on Immunization Practices. They met on a Saturday, December 12th, to review the information and the FDA Emergency Use Authorization, and they came out with their recommendations for dosing, for time frame, and for monitoring, and for who should and should not be offered this immunization. Once that was done, the whole world was paying attention to this, and once the CDC Advisory Panel produced their recommendations, Hawaii, of course, just like every other nation across the U.S., wanted a piece of it. So this is the Hawaii use. Our first vaccine, since the manufacturer was already developing this, boxing it up, and once the approvals came through, they were ready to ship it out. They were letting the state of Hawaii know that they were going to be shipping out vaccine sometimes just as soon as the committee made their recommendation. We received the first doses of the Pfizer vaccine on December 14th. This clip here from Hawaii News Now was a video that was shot when the vaccine actually arrived at the Queens Medical Center punch bowl and was stored in our ultra cold freezer. Some of the background work that had to be done was to identify and procure these ultra low temperature freezers, which basically is colder than the freezer that we would normally have, say, in our house or in our pharmacy. These are ultra cold storage and are down to the temperature of dry ice. The state of Hawaii actually was very good as far as for trying to use the resources that we had at Queens West Oahu. We were actually lucky enough to have partnered with the University of Hawaii, and one of their ultra cold freezers they've actually donated to us, and we have in our facility, and that is what we are currently using. We will give it back to them, and we and the entire West Oahu population should be very thankful to the University of Hawaii for sharing that resource for the benefit. The first vaccine was administered at Queens Punch Bowl on December 15th. And for a lot of the healthcare workers, this has been a whole year at the end of the year, near the holidays, and the United States has been dealing with COVID at that point for about 9 months. And this was all with just the idea of treating it once people got infected. This was something that we really were looking forward to because it could help stop the COVID-19 syndrome or virus from affecting our population. However, being a limited resource, the state had to figure out, well, who's going to be getting this? Because the fear, being locked down, being inside of the house, having our children have to learn from home, a lot of people really wanted to get it, especially the folks who were looking in the research and trying to figure out if it was the right thing for them. Once people were able to process the information and find out if they wanted it or not and make their own personal decisions, a lot of people really wanted to get access to it. However, the supply that was coming week by week, and it couldn't give access to everyone. So the state of Hawaii did partner with a lot of the large healthcare organizations for expert advice. They actually asked us what we thought and how we thought that allocation and distribution of a very scarce resource would benefit the people of Hawaii the best. It did roll out into different phases, and it did follow along with some of the general guidance that the CDC came out with, which was in the phase 1A in the beginning, give it to the healthcare personnel who are going to be taking care of the patients who come down with COVID, and allow the people who would be caring for sick people to have some protection so they can continue to treat people once they're sick. Also, the most vulnerable, the long-term care facility residents, were in that grouping. Then we got down to the phase 1B, which was the kapuna, the age 75 and older, as well as the frontline workers who were essentially put out there to continue their work and expose themselves to the general population for where they would come in contact with people who had COVID, especially the firefighters paramedics, frontline workers, people who had to be in close proximity to people, food line workers, and those types of police officers. And the column on the right here shows how much of the population would be covered by all of that. To get through phase 1 and phase 1A and phase 1B, that's basically only about 25% of the population. So still, that was a large chunk, but it was a scarce resource. What we really wanted to do was avoid what we were seeing in some of the cities on the mainland. California was using Disneyland's parking lot as a mass vaccination site. And you can see here that it was out in the open, it was exposed, it was kind of a free-for-all walk-up. And the whole concept of social distancing for safety, when we were in Hawaii looking at this, we were thinking that, yes, you want to provide vaccinations, but you don't want to make people in more danger than they needed to be. So what we ended up doing and recommending for a lot of our mass vaccination in the phase 1B was a sign-up system. Because we're looking at essential workers and we're looking at Kapuna, we wanted to have an appointment system so that if you're 75, 80 years old, 85 years old, and you needed to come to a center, that you wouldn't be standing in line for 3 hours with somebody else who's 45 years old. Also, just the amount of energy it takes for one of those folks to stand up and stay in line is draining. So we did partner with some of the community members with HMSA, with Hawaiian Tel, and we set up telephone lines. So people who did not have access, if we're in rural communities or even did not have large data plans, could call a telephone number and sign up on our websites. Because we were still in the 1A, we trusted people and we needed them to self-attest that they were in those age brackets or in an essential worker population. But after that, we really believed in the people in Hawaii and wanted to say that if you signed up and you tested and you said that you were part of this category and it was your turn, that you would then take your time in line and you would wait if it wasn't your turn yet. We would be checking ID, but at that point, we wanted to make sure that we allowed people to have access. Queens opened up its Blasdell mass vaccination site for phase 1B on January 23rd. So again, that timeline is still very, very short between when we were able to start vaccinating the phase 1A and to start vaccinating the general community. The Blasdell's been a huge success. Queens as a system has vaccinated over 100,000 people, put that many vaccinations into people's arms. And at that point, the vaccine supply increased. So we were very happy when the state allowed us to have a larger supply of vaccine and open up our West Oahu campus for vaccination. Even when we were just announcing that we were going to be opening it up, it did make the news. KHON, the Star Advertiser, covered the announcement that we were going to be opening up a West Oahu mass vaccination site. And it did just reflect what we were hearing from our partners in the community, as well as just the people that work here and live here, that having something here at West Oahu would be a huge win for us. We did a lot of preparation. One, we did have the ultra low cold freezer, and we did have a soft opening for the phase 1A workers, not only here at Queens, West Oahu, but also the doctors, the dentists, the therapists out in the community to come to Queens West and get their vaccinations if they're phase 1A. But when you're talking about the overall population, the kapuna, the elderly, we really need to set it up a system where it would be streamlined. We did a lot of training with our staff as far as for wayfinding. Healthcare providers are comfortable in hospitals. When you talk about people in general, usually you'll come in for your appointment and go. Nobody really wants to hang out at the hospital like we will at the beach or at our cousin's house or something like that. So we noticed that streamlining the process and giving direction was very important. Actually similar to the Disney experience where you start at one point and you just continue in a straight line through. Also, quality was very important. Our pharmacists are on site and they are helping us mix the vaccine itself, and make sure that we're getting all the correct dosages out of it. Our staff had to do a lot of training to get themselves prepared for registering people into the system, because not only are we going to be administering the vaccinations, but we will also be keeping up the Queen's quality of monitoring people for any side effects and reporting into the national system for any adverse events or hospitalizations or emergency department visits after people get vaccinations. That also helps contribute to the knowledge that we will have of the efficacy of these vaccines. We still are in the phase 3 trials where the long-term effects of the vaccines, both the preventative as well as any adverse events, are being monitored. But this was the day that we were all excited for, the day that we were able to open up our phase 1B vaccination, the phase 1C vaccination to our community. Here is a picture outside of our clinic of Mr. and Mrs. Kiani, who are our first two kapuna to receive the vaccine at Queen's West Oahu. This is a picture of Mr. Kiani getting his vaccine. We do know that when we asked him, you folks actually were able to get your vaccination. Why'd you wait to so long? Or, it wasn't a long time, but not go down to the Blasdell or the Pier 2 when you had the opportunity. A truly Hawaii answer was he wanted to wait his turn. He and his wife wanted to wait their turn, and that they didn't want to go down to the Blasdell. This is a photo of one of our infection prevention doctors, our infectious disease doctors, Dr. Heath Chung, getting his vaccine earlier. And of course, I want to recognize the fact that our doctors are our neighbors, and they put themselves at risk to take care of people who actually came down with COVID. So having them being able to become protected as they're putting themselves in danger was also very important to us. Photos of our grand opening. We did a blessing of the clinic to bless and protect all the people who would come through, as well as who worked within the clinic. And our local and national representatives did want to make sure that their constituents were protected. So this, on the bottom right, is a photo of Representative Case coming down to the vaccination clinic to see what we were doing and make sure that the people that he represents are taken care of. He had a very big interest in the Native Hawaiian and Pacific Islander community. And from our data, we're actually able to see that of all the people that are signing up and coming through our doors now, over 10% of them are identifying themselves as Native Hawaiian and Pacific Islander. This is just a snapshot of what we're looking forward to even on this weekend, of how many people are coming through. The red bar are the people who are coming through our West Oahu clinic per hour getting their first doses. Blue bar are people who have come through in the last 3 weeks and are coming in for their second dose. So this is the last portion of the presentation that I want to do, which is to talk about the science behind the vaccine, because people are asking not only about the timeline, but what is this? I know about the tetanus vaccine, I know about the flu vaccine, I know that there's been jokes in the news in the past about the flu vaccine not being the right target. So what does it mean about this Pfizer vaccine? So going back to what we were talking about earlier, the technology for this messenger RNA approach to vaccine was actually developed over 15 years ago in 2005. Developed in the United States, it was using a code that is seen as the genetic code of the virus and modifying it. So when people talk about their genes, viruses also have genes, but they're a very primitive organism, and their code is within them. And what the technology did was it took a certain segment of that code or the programming and only took that portion of it and targeted that. Currently, that technology is also being used to develop vaccines for other things, including genital herpes, influenza, and HIV. So this is not a new technology that was just discovered and developed in 2020. It actually has been out there around, but from that other graphic showing how long it takes to develop because of the cost constraints, because of the different staging, it will take longer than how it was approached and everything was done at the same time. So the vaccine is developed in a way to protect the messenger RNA code because the body is actually terrific. Normally, our immune system will break down anything that is injected into the body, which is a messenger RNA, so quick that we would not be able to use it in an effective way. The development of this messenger RNA vaccine by Pfizer, it actually used the coating, but it protected it in a lipid envelope. Lipid means like a fat or a wax. If we're looking at waterproofing something, if we're going to use an example that is not for this vaccine technology, but just in keeping our things safe when we're going camping, you protect it in something that protects it from the elements. In this case, the elements would be our natural body immune system. The lipid envelope temporarily protects the messenger RNA message so that it can get into our body and do what it's supposed to do. However, the lipid technology is very fragile. It doesn't last very long, which required that ultra cold temperature for the storage. This is a graphic, a cartoon from the CDC. It's a big, busy picture, but what it does is it kind of walks through from up here, going down through the system, and what it does to protect. So I'm going to break this down a little bit for us. In this stage here, this big picture is a breakout of this area right here. This is a picture of the vaccine in a cartoon fashion. They take the messenger RNA, which has the code, only a small segment of the actual COVID code, and they put it into this lipid nanoparticle. And as this box here says, the vaccine does not contain any virus, so it cannot give you COVID-19, and it cannot change your DNA in any way. The scientific description of it is here at the top of this slide, saying that it's a lipid nanoparticle formulated nucleoside modified RNA vaccine that encodes a pre-fusion stabilized membrane anchored SARS-CoV-2 full-length spike protein. And the key thing is that it only codes for the full-length spike protein, so a small portion of the COVID vaccine. That then is injected into the recipient or the patient here. And what it does is the code does not go into what we call our DNA area, but it does go into some of our cells and uses what's called a ribosome or a protein making portion of our body. It takes that small segment of the spike protein code and it produces that protein, and it allows our body to see it and says that it's foreign, and our body attacks it and forms antibody from memory. That spike protein then, in case we're ever exposed to the SARS-CoV-2 virus itself, is already primed and prepped. So if this actual virus itself gets into our system because we're exposed by somebody else who had COVID, these antibodies are ready and waiting to help prevent and protect us to fight it off. This last portion of it is just a smaller picture saying that after the messenger RNA delivers the instructions, our cells, our antibody cells, will break it down and get rid of it. This is a little cartoon of what we're actually looking for. When we have a bad guy or a robber in this case, and you're looking at a virus, you don't want to let the whole robber into your squad room when you're training your police to identify and arrest these people. You would not want to have them around anybody else. You actually want to identify them early and prevent them. What the vaccination actually does is it actually looks for something that is only identified as the bad thing, that spike protein. So if we're going to extrapolate it, it's kind of like if we trained the police officers to look for a specific type of mask. If you see the mask, those are usually the bad guys, and you want to kind of go after them and arrest them. And it is a new technology for vaccines, but it seems to be incredibly effective. Again, this is the article that was published in the New England Journal, and I'm going to break this down. What it did was it it let us know what Pfizer had actually done with their research. They did a worldwide test of 152 sites. 76% of these sites were in the United States, but they also did other countries, including Argentina, Brazil, and South Africa. They published the results of their phase 3 studies, and their participants were people who were 16 years of age and older. This is one of the key reasons why, when we're talking about the different age ranges, only the Pfizer vaccine at this point can be used for patients that are 16 to 18. The Moderna research hadn't gone down to the minors or published that information, but the Pfizer vaccine did give out that information. So they enrolled 43,000 people into this study. And in this study, they separated they randomized it, and it was pretty equal between men and women. 42% of the people were over 55 years old. 83% of the people were white. 9% were Black or African American. 4% were Asian, and 2.3% were multiracial. And in this study that they used, 76 of the participants were identified themselves as Native Hawaiian or other Pacific Islander. You know, in Hawaii, we would really like to have a larger population, but this was the study, and this was the population that they actually used. The Pfizer trial actually was done in Hawaii as well, having one of the sites. So it does make us in the medical community here in Hawaii a little bit more comfortable because it was done in our backyard. So what they did was they separated the two groups. 21,000 people received the vaccine injection, and 21,000 people received the placebo. In any good scientific study, you want to see all the information because it won't be perfect and not everybody will complete the entire thing. So in each group, about 3,000 people did not complete the 2-series injection. What did the results show? For the placebo injection here, this is an injection that did not have any of the COVID vaccine in it. What they did was they immunized both groups, 21,000 people each, and then they monitored them afterward. And they had the same exposure to just the general world. And what they found was that in the general population, after 7 days, if you're exposed to somebody who's had COVID or not and just doing your general work, 169 people actually tested positive for SARS-CoV-2. And this includes people who are symptomatic as well as asymptomatic. In comparison, they took a look at the people who got the immunization. And of those people who had the same behaviors, same exposures, same lifestyle, they found that only 9 people came down with the infection. And of course, this I want to point out is with normal behavior. So in those sites, the over 100 sites across the world, whatever their local protocols were, masking, physical distancing, that was what they were practicing. That translates into the finding that the vaccination is 95% effective in preventing COVID from people who do not have vaccination, but also still have their normal regional precautions for SARS-CoV-2. So what are they looking for when they're trying to find out if the FDA will give an authorization for a vaccine to be brought to the general population? Their thresholds were actually set at 50% effective. When we do talk about one of the years previously where the influenza vaccine was not as effective as we thought it would be, that effectiveness rate was down in the 30% for the circulating virus at that time. But when you get a threshold of 50% and the Pfizer vaccine was 95% effective at preventing people from coming down with COVID, that is a huge win. It is something that in the medical field we're very happy with, we're comfortable with, and this messenger RNA technology appears to be working incredibly well. The second question that people are going to ask is, well, how long does it last? Will I have to do a booster shot? What's the threshold? That information, it is still too early right now to have a definitive answer, but in this clipping here from the medical literature right now, they tested to see what the response was from people who actually had COVID and people who got the vaccine. They measured the antibody levels. Science at this point does not know yet what the threshold is for a good level of antibody to have in a blood test to say that, yes, you're very well protected. However, what they did do was they tested to see if the vaccine had people generate a higher level of antibody to the spike protein than somebody who's had a prior COVID infection. And they did find that with the two-stage vaccine, the antibody level in the blood is incredibly responsive and actually was higher than somebody who had gotten COVID in the past. People who got COVID in the past and then were given one dose of the vaccine had a larger antibody circulating in their bloodstream. So that lets us know that the vaccines are actually better at eliciting a vaccine antibody response than just being exposed. So this herd immunity concept is good. People will have immune memory and they will produce antibodies if they come down with COVID, but they will have a larger antibody production if they get the vaccine. So that is part of the information that we are using to base the recommendation that everybody, even if they've gotten the COVID infection, get their COVID vaccine. This journal article on this letter on the right is very hot off the presses, and it was submitted just last week, Friday, on the 9th of April. And it is not the Pfizer vaccine, but it is the data from the Moderna vaccine. And it's a similar technology, and it describes what they're finding in their follow-up studies of people and their blood tests who had received the Moderna vaccine, their two-step vaccine. And 6 months after their second vaccine, they're still finding a good level of antibodies in the bloodstream. It does note that the level of antibodies detected in the bloodstream actually decreases as you get older. It is still a higher level than people who are exposed to COVID, but it does decrease. So people who are younger will produce more antibodies after vaccination than older people. So that does have promising information, and we'll continue to monitor and look at the research that's being done with the vaccines and from their manufacturers. So that is the information that I thank you for allowing me to present to you. And I'll hand it back to Eric if there are any questions that any of the participants had.
Thank you very much, Dr. Ronald Kuroda, for the very informative presentation. And really, thank you for your leadership and all the exceptional work that you've done for the pandemic. Yeah, so right now, let's proceed with the Q&A or question and answer portion of our presentation. Does anyone have any questions? I'm looking at the chat box and we don't see anything, but we can give you maybe 10 seconds or so if you have any last minute questions you want to ask Dr. Kuroda.
I think I do see a question here in the chat box.
Let's see.
Are there any updates on the pause of the Johnson & Johnson vaccine? Okay, good. Thank you for asking that question. It is a very timely question. The recommendations that have come out from that same CDC panel is they don't have enough information yet. The cases that have come up, the millions of Johnson & Johnson vaccines that have been administered, approximately 2 weeks after the administration, 6 women have come down with what's called a thrombosis in one of the blood vessels of the brain, and they also see that their platelet counts drop. 6 out of millions, about 7 million doses that have been administered in the United States, is a very low number. Actually, if you do that math, it's about 1 in a million. But they don't have enough information yet to make a solid recommendation one way or the other. One of the things that we do talk about in science is it a causation that you see something happening, or is it an association? One of the examples that I like to use in Hawaii is we all know that it's very sunny outside, and every once in a while you'll be walking around and it'll be sunny and it'll rain on you. If you looked at it and you just took one snapshot at that time and you said, "Oh, I'm getting rained on and it's sunny," one thing that can be drawn, and what you would have to test that hypothesis is, does that mean that when it's sunny, it rains? And that would be causation. Or is it just that there's an association of the observation? I think that we can all see that you would need a rain cloud above us to get wet and for there to be rain, even though it's sunny. So from that example, it would not be causation. And at this point, we're still going to have Johnson & Johnson follow along. We are monitoring and reporting any of the side effects or adverse events from the Pfizer and the Moderna vaccines. And we're taking a pause on the Johnson & Johnson, but nationwide, we'll watch if there are any other adverse effects from the 7 million doses that were administered. Thank you for that question.
Yeah, that was a great question. Any more questions? Look like there is none. And again, thank you very much, Dr. Corder, for making things so easy to understand. I think that's the reason why we don't have that many questions today. But again, thank you. And of course, to all of you, our virtual audience. And that concludes our Speaking of Health presentation for April. Again, the West Oahu Virtual Speaking of Health series is the third Thursday of each month, same time, 12:30 to 1:30 p.m. Tune in for the next lecture, which will be coming to you on Thursday, May 20th. And our speaker will be our urologist, Andrew Sun, who will be talking about kidney stones and you. Again, his topic will be kidney stones and you. So look out for the announcement, which will include registration information. We hope to see you then, and feel free to share or forward the announcement to your friends, family, colleagues, and anyone you think may be interested. On behalf of our COO and Senior Vice President Susan Murray, our Chief Medical Officer, Dr. Ronald Kuroda, and the entire Queens Medical Center West Oahu, mahalo nui loa. Thank you very much for joining us today. Until next time, please take care and always be safe.
And wear your mask, please. We're almost out of this. Be careful.
Thank you. Have a good day, everybody.